Good morning, everyone. My name is Linah, and today me and Ahmed will be explaining the citric acid cycle. So we understand that biochemistry, it's not like bio, I understand that you would prefer a more detailed explanation kind of a session rather than a revision, especially since you just began this chapter. We're going to try to give you the best tips and tricks to remember. We're going to give you questions, and at first it might not make sense, but as we go ahead with the semester, we will start to link everything together. And I know everyone's been saying this to you, but I promise you it's true. Like by the time of the midterm, everything will come together for glucose metabolism, carbohydrates, and then after that we'll take lipids, amino acids, and it will come together as one big picture. So for context here, first I will be giving you a revision on redox reactions and energy, like as a whole, like oxidation, reduction. Then we will be linking glycolysis to the citric acid cycle by the link reaction. Then I will begin with the citric acid cycle, and then Omar will continue with it. And finally, citric acid cycle regulation. Okay. So first of all, reactive oxygen species. We have molecular oxygen, but it may form toxic oxygen ions and peroxides called reactive oxygen species. And these may react with, damage, and destroy biomolecules. The cells can cope by two ways: detoxifying enzymes and antioxidants. Now, this isn't really the main point of the chapter, but it's important to know at the back of your head. So we have organisms can be classified based on how they use oxygen for energy generation, or whether they use it or not, and for their tolerance to reactive oxygen species. So we have obligate anaerobes. These do not use oxygen, and they use fermentation to produce energy, and they cannot tolerate reactive oxygen species, meaning that they cannot survive in any environment with oxygen. Then we have aerotolerant anaerobes, which are tolerant to reactive oxygen species but don't use oxygen for energy. Then facultative anaerobes, which usually don't use oxygen, but if oxygen is available, they may use it to produce extra energy, and they're tolerant to reactive oxygen species. And obligate aerobes, which are humans, animals, which require oxygen for survival and have developed like multiple antioxidants, detoxifying enzymes, and things to be tolerant to reactive oxygen species. Okay, so aerobic metabolism as a whole. So far you've done glycolysis, which is anaerobic metabolism because it does not require oxygen. So you've done pyruvate. Now we will be converting pyruvate to acetyl-CoA, and then there are 3 steps to aerobic metabolism, which require oxygen. So as you can see here, this basically is a summary of basically all of biochemistry for this semester. You will have glucose, fatty acids, and amino acids. All of them produce acetyl-CoA, which will go into the citric acid cycle and therefore produce energy. So quick comparison. Anaerobic metabolism includes glycolysis, and it happens in the cytoplasm. Then once we enter the mitochondria, we have aerobic metabolism consisting of citric acid cycle, electron transfer chain, and oxidative phosphorylation. Now, anaerobic metabolism, the issue with it is that there is less ATP produced overall, but it is produced faster and oxygen is not required. As for aerobic metabolism, more ATP is produced. Okay. More ATP is produced, but it does take more time to produce the ATP, and oxygen is required. In anaerobic metabolism, there is incomplete oxidation or breakdown of glucose, as glucose is only broken down from a 6-carbon compound to a 3-carbon compound, which is pyruvate, and then we stop there. But for aerobic, we continue until we have just carbon dioxide and water. Okay, so redox reaction. This is a general rule that you should always have in the back of your head, which is that whenever you see gain of oxygen, loss of hydrogen, loss of electrons, or increase in oxidation number, that means you have oxidation. And the opposite is true for reduction. So when we are dealing with energy, we have redox reactions. So you will see continuously oxidation because we are producing energy. To release energy from a nutrient molecule, we oxidize them. Okay, so for example, in metabolism, we will keep oxidizing over and over until we release the energy. And we keep losing electrons, which equals loss of energy. Whenever you see loss of electrons, it means energy is released. The opposite is true when molecules gain energy when we reduce them. For example, in photosynthesis, we reduce carbon dioxide to glucose. So it gains energy by the reduction. So you should always have this rule at the back of your head. And electrons are the intermediates in redox reactions. The tendency of a specific substrate to gain electrons is called reduction potential, which means that when when a molecule has a greater reduction potential, it means it has the greater potential to be reduced or gain electrons. So when you compare two molecules, the one with the higher reduction potential has the higher affinity for electrons and gains the electrons. Okay? So higher, more positive reduction potential is equal to higher affinity to electrons and it becomes an electron acceptor. The opposite is also true. Lower, more negative reduction potential means that they're more likely to lose the electrons. They don't have a high affinity for the electrons, and therefore they're an electron donor. Electrons are transferred from a redox pair with a lower, more negative reduction potential to those with a higher and more positive reduction potential. And this you will see as questions where they will show you the reduction potential of two different reactions. And you choose which one will gain electrons and which one will lose electrons. Okay? Okay, so for example here, you will see this is actually one big continuous table, but I split it into two slides. You can see that the reaction or the redox pair with the lowest reduction potential is hydrogen. This means that hydrogen will always lose its electrons or donate its electrons. As for here, it's all increasing until you reach oxygen, which has the highest reduction potential. What does this mean? It's more likely to gain electrons and be reduced. Which makes sense because oxygen, when we put it with any other compound, it will always oxidize the other compound and be reduced because it's oxygen, it will oxidize. And this is how we know that oxygen is the final electron acceptor. Of course, you will go into more detail into this in the electron transport chain and oxidative phosphorylation. You will learn that this is a direction on this is the direction of electron transfer. Electrons will move from molecule to molecule in the electron transport chain until they reach ATP synthase and produce ATP. So oxygen is the final electron acceptor. How do we know this? Because it has the highest reduction potential, and this is why aerobic metabolism cannot take place in the absence of oxygen. Okay. Is the following reaction a redox reaction? If yes, identify the oxidizing and reducing agents. So who wants to do the first one?
It's not a redox reaction.
Yes. Here we have, actually it's a phosphorylation. You can see that the phosphate is transferred from ATP to glucose. So would you like?
No, thank you.
No?
No.
Okay. And for the next one?
It is a redox reaction.
Yes.
NADH got oxidized.
That's true. Because it's lost its hydrogen. And then CH3CHO got oxidized. Reduced because it gains the hydrogen. Yes. Okay. Which of these reactions will receive electrons? So this is actually, this question, we can see it a lot, and you ignore the entire equation and you focus on the reduction potential. So which reaction will receive electrons? Yes.
Yeah, positive 0.42.
Yes. Because it has the higher reduction potential. Exactly. Okay. Can you give up? Okay. actual citric acid cycle, you will see these redox coenzymes multiple times. So we should go over and over for them. First of all, nicotinic acid, which is a vitamin, and it has two coenzyme forms. That means it's activated to form these two coenzymes, NAD and NADP. They're basically the same compound, except NADP has an extra phosphate group. So the basic structure is adenosine with an N-ribosyl derivative of nicotinamide. This is the adenosine. This is the nicotinamide. And they're joined by a pyrophosphate group. And for NADP, it would have an extra phosphate group on the 2' hydroxyl group. This, you need to know it. It will help you imagine when the reduction and the transfer of hydrogen happens. But you don't really need to memorize it. So for nicotinic acid, the function, we have both NAD and NADP can act, they are electron carriers for dehydrogenases, which are enzymes for redox reaction. So basically, when we have a redox reaction, we will need the NAD and NADP will act as reactants, and they can act as oxidizing agents, and NADPH and NADH act as reducing agents as they donate their hydrogen. NAD specifically is used for catabolic reactions, as you will see in energy generation, any sort of breakdown, you will keep encountering NAD. NADP is used for anabolic reactions where we have biosynthesis of molecules, so you will see them in later chapters. And finally, NADPH is an antioxidant to protect from the reactive oxygen species we mentioned in the beginning of this session. Then know that NAD and NADP are not part of the enzyme. They act as separate reactants or coenzymes. So they bind transiently, and after the reaction, they are separated. As for the second one we have is riboflavin, which is vitamin B2, and the two coenzyme forms are FMN, FAD. This is the basic structure. You have riboflavin, which has an isoadenosine ring system, and for FMN we have one extra phosphate group. For FAD we have two phosphate groups and an adenosine. And this group acts as a donor acceptor of two hydrogen atoms. So in NAD, it can only accept one hydrogen atom. Here, you will see that it becomes FADH2, which means that it's a donor acceptor of two hydrogen atoms. And it functions, both of them function as electron carriers for a group called flavoproteins, which are enzymes that work with the riboflavin. And here it's not transiently bound, it's a prosthetic group. So FAD will stay with the enzyme it works with. After the reaction, it will stay on the same enzyme and it doesn't diffuse away as a reaction, as with NAD. And for two electron transfer reactions, we can see them in the mitochondrial matrix, and FAD is ideal for this as it accepts and donates two hydrogen atoms. And if it's a one electron transfer reaction, as in the electron transport chain, it will work by transferring one hydrogen atom at a time. Okay, so now that we're done with this part, this is a quick recap on glycolysis. As you can see, we've already done this. It's the basic equation of glycolysis. So the key points to remember from this, especially today, is that the net ATP produced is 2. Everyone knows why this is, right? Why is it net ATP 2?
Because it consumed 2 ATP in the beginning and then it produced 2 more afterwards.
Yeah. It produces 4 more. Yeah. It consumes 2, produces 4, so in total you have 2 new ATP. Okay? And also we have we have 2 NADH produced. Does anyone remember which reactions the NADH was produced?
6.
6, yes. And the glucose, which is 6 carbon atom, is broken down into 2 pyruvates, each with 3 carbons. So these are the key takeaways you need to remember from glycolysis. The next reaction, this is the first reaction where we require oxygen to work. So it doesn't actually require oxygen as a reactant, it just requires the presence of oxygen for the reaction to work. Acetyl-CoA is an acetyl group from carb fatty acid amino acid linked to an acyl carrier molecule called coenzyme A. So this is coenzyme A. It's just a carrier molecule. Then you have the acetyl group, which is a group of 2 carbon atoms that link to this coenzyme on the reactive thiol SH group. Okay? So when we have an acetyl group of 2 carbons, it will link on this thiol group and the end. Okay, so the link reaction takes place in the mitochondrial matrix, and its type is oxidative decarboxylation. This is because we go from pyruvate of 3 carbons to acetyl-CoA, which has 2 carbons. So we've lost a carbon, it's decarboxylation. And it's oxidative because the reaction is an oxidation reaction. The enzyme we require is pyruvate dehydrogenase complex. This isn't a normal enzyme, it's actually a very large enzyme with many enzymatic activities. It's incredibly large and it's found in the mitochondrial matrix. This is an irreversible reaction and it goes in only one direction because why is it irreversible? We don't have the enzymes that facilitate the reverse reaction, and the reaction is highly exergonic, meaning that it releases energy, so the forward reaction is favored. This is the basic equation: pyruvate plus NAD plus coenzyme A. By the way, we write it as coenzyme ASH because it has the thiol group, which isn't carrying an acyl group yet. To produce acetyl-CoA, NADH plus H plus, and 1 carbon dioxide molecule. So yeah, large multi-enzyme complex, 3 enzymatic activities. The first one is E1, pyruvate dehydrogenase, then E2, dihydrolipoyl transacetylase, and E3, dihydrolipoyl dehydrogenase. And we will take each activity and how it functions. And finally, it also requires 5 coenzymes. So I'm going to start by reading the coenzymes and their function. Then I'll be explaining the reaction in detail so you can see where these coenzymes are used. First of all, thiamine pyrophosphate, TPP, which wherever you see this compound, know that it's a decarboxylation. So this will facilitate the removal of the 1 carbon molecule and the production of carbon dioxide. Lipoic acid, which is a carrier of hydrogens or acetyl groups. In this case, it will be a carrier of an acetyl group. So it will remove the 2 carbon group and donate it to coenzyme A. NADH, FADH2, they're both electron carriers, as we mentioned. And coenzyme A, which is an acyl group carrier. Okay, so this might look a bit overwhelming, but this is exactly how you should remember this reaction. So I will teach you how to draw it step by step, and then wherever you need to remember this reaction, it will take you 2 seconds to draw it. Okay, first of all, we're going to start the reaction with 1 pyruvate and the TTP coenzyme. Okay, thiamine pyrophosphates. Okay. So the first enzymatic activity, E1, which is pyruvate dehydrogenase. So what happens here is the decarboxylation reaction. So what happens is we have a 3 carbon compound here. 2 carbons will be transferred to TTP to form hydroxyethyl TTP, and the 1 remaining carbon will form carbon dioxide. So this is what happened in the first enzymatic activity. Now, there is some explanation on the mechanism, which you should read as it will help you to understand what goes on, but usually it's not really asked of us to know this. So pyruvate is decarboxylated by pyruvate dehydrogenase. The thiazole ring of TTP is deprotonated by a basic residue of the enzyme, forming a nucleophile. This nucleophilic thiazole ring attacks the carbonyl group of pyruvate, forming hydroxyethyl TTP and carbon dioxide. This carbon dioxide, by the way, is released. So now it will be released and excreted by the lungs. So the concentration of carbon dioxide will be very low. This will force the reaction to keep going forward. TTP needs to be returned to its original form to be reused. Yes, so now we have hydroxyethyl TTP. We need to return it back to its original form so that we can decarboxylate the next pyruvate that comes. So what do we do now? We have lipoamide, which is the next coenzyme. This is the second coenzyme we will use. So hydroxyethyl TTP will donate this 2-carbon hydroxyethyl to lipoamide. This will produce TTP and hydroxyethyl lipoamide. This happens in E2, which is dihydrolipoyl tranacetylase. Okay, and this is an ethyl transfer. It's as simple as that. So basically, you take this HE and you give it to lipoamide. So now lipoamide is a temporary carrier of this hydroxyethyl group. So we need to transfer it to coenzyme A to produce acetyl-CoA. So this is what happens. Okay. Coenzyme A, okay, the hydroxyethyl group, the 2 carbons are transferred to coenzyme A to produce acetyl-CoA and dihydrolipoamide. So now we've done what we wanted to do in this reaction. We've produced carbon dioxide and mainly we've produced acetyl-CoA. So we've done everything we need to do. There's only one issue remaining, which is that dihydrolipoamide is in its reduced form and it needs to return to lipoamide in the oxidized form, which is active so that the reaction can keep going. So what we do here is we need to reoxidize lipoamide, and we do this by FAD. So FAD will accept the hydrogens and it will be reduced and dihydrolipoamide will be oxidized again. So we will have FADH2. Okay? And this happens in E3. And it's just a redox reaction. Now we have one last remaining issue. FADH2, as we mentioned before, it's a part of the enzyme. It's still attached to the enzyme, and now the enzyme can't work if it doesn't have its original oxidized FAD. So what we will do is we will have NAD. NAD will oxidize the FADH2 to produce NADH plus H+. So now it's not a problem. The NADH will go and it will be reoxidized in the electron transport chain, and we can get a new NAD. And now the reaction can keep going. Now there is one exception in this reaction. Usually, NAD plus is more. It has a more negative reduction potential than FADH2, which means that it's usually the electron donor. But here, as you can see, it was the electron acceptor. So this is because FADH2 is part of this enzyme which alters the way it works. So this is just a fact that you should know. But this is how it works. But usually, NAD donates electrons to FAD. NADH donates electrons to FAD. Okay, by the end of this reaction, what do we have? We have per pyruvate, 1 acetyl-CoA produced, 1 carbon dioxide, and 1 NADH. Now we have acetyl-CoA, it can go directly enter the citric acid cycle. Okay, as a revision, pyruvate dehydrogenase. The function was to decarboxylate pyruvate using which coenzyme? TTP. Dihydrolipoyl acetylase, E2. The function was to transfer the acetyl group to coenzyme A. So first it was transferred from hydroxyethyl TTP to hydroxyethyl lipoamide and finally to acetyl-CoA. And then we have E3 dihydrolipoyl dehydrogenase, which will reoxidize the dihydrolipoamide using FAD. Okay? And we have multiple, as you can see, we have many coenzymes here. that require to go back to their original form. So you need to remember how each of these is returned to their original form. TTP is we need to return it back to hydroxyethyl TTP back to TTP. So we do this by lipoamide. Lipoamide accepts the hydroxyethyl group. For lipoic acid or lipoamide, it's reoxidized by FAD of E3. FAD is reoxidized by NAD+, and NAD+, you will take that, it will be reoxidized by the electron transport chain. So all of these coenzymes are regenerated by these ways. Okay, now for questions to test their understanding. Which of the following statements describes the function of lipoic acid during the conversion of pyruvate to acetyl-CoA? Should I read the options or would you like to read? Okay.
B.
B. Reacts with hydroxyethyl TTP to produce acetylated lipoamide. It's correct, but I want to check if it's correct. Yes, it's B. Yes?
Is it acetylated lipoamide or ethylated lipoamide?
Hydroxyethyl TTP to produce acetylated lipoamide. So here we're talking about a 2-carbon compound. So generally, when we're talking about lipoamide, we usually use acetylated because it's a 2-carbon compound, and this acetyl group will be further transferred to coenzyme A to produce acetyl-CoA. But when it's with TTP, it will be called an hydroxyethyl group because it just depends on where the atoms are attached on the coenzyme. Okay, a deficiency of vitamin B1 thiamine was most Okay. A deficiency of vitamin B1 thiamine will most directly impair which step of the link reaction? So where do we need vitamin B1 thiamine? Which is TTP, by the way. Yes?
C.
C. Decarboxylation of pyruvate. That is correct. Yes, this is the function of wherever you see vitamin B1, wherever you see thiamine, it's always decarboxylation, and you will keep re-encountering this over the course. Okay. Which of the following best explains why the link reaction is considered irreversible in humans? It occurs in the mitochondria, it requires multiple enzymes, it releases a large amount of free energy, or it produces carbon dioxide. C. It releases a large amount of free energy. That's correct. Wherever you see a reaction with negative delta G or it produces a lot of energy, this means that it will be irreversible or difficult to reverse. Okay, finally, a toxin inhibits lipoamide reoxidation in the pyruvate dehydrogenase complex. Which product will decrease first? Yes?
B.
B. NADH. Okay, so let's look back at what lipoamide reoxidation. So here we have lipoamide. Imagine it's not reoxidized. Which product will decrease first? Yes, it's B. NADH. Because we will not have FADH2, that means we cannot produce NADH. Actually, other compounds from these options will also be decreased, but the question is asking which will decrease first. The keyword is first. Okay, so regulation of the link reaction. This reaction is strictly regulated. It's strictly regulated because this reaction is it has a very important function in energy production. So when we have low energy levels, it will be activated to produce energy. When we have high energy levels, it will be inhibited because we don't want to produce too much energy. This is how the body works. So it's regulated by two methods. First of all, allosteric regulators, which are compounds that bind to the enzyme to activate or inactivate it. So it's activated by AMP NAD plus coenzyme A. So personally, I never ever memorized, okay, AMP activates this enzyme. I never memorized this blindly. What you need to look at is what the compound means. So when you look at AMP, what does it mean? It shows that we have low ATP levels, or AMP signifies low energy levels. So logically, it will activate the pathway because it will lead to energy production. As for NAD plus and coenzyme A, they are substrates of or reactants of this reaction. So when we have more NAD and coenzyme A, the reaction will be activated. And the opposite is true. When we have ATP, it's a high energy indicator. NADH, acetyl-CoA are products, so this will inhibit the reaction, or it will inhibit the enzyme. And another way for regulation is covalent modification, which means that we will have the enzyme's covalent structure will be modified for it to not work or for it to work better. So usually when we see phosphorylation, it means inactivation. So here we have acetyl-CoA and NADH, as we mentioned, they are products, so they will activate pyruvate dehydrogenase complex kinase 1, which will phosphorylate the enzyme, and when it's phosphorylated, it's inactivated. Pyruvate, coenzyme A, and NAD are reactants, so they will inhibit this kinase, therefore we will not have phosphorylation, and pyruvate dehydrogenase complex will remain active. And finally, when we have low mitochondrial ATP concentrations, calcium and insulin, we will activate another enzyme called pyruvate dehydrogenase phosphatase. So phosphatase, when you see kinase, it always means phosphorylation. When you see phosphatase, it always means dephosphorylation. So here we will dephosphorylate the previously phosphorylated or inactivated pyruvate dehydrogenase complex, and therefore reactivate it. Okay, so as for the rule I mentioned earlier, for not blindly memorizing, this doesn't work here. You have to remember that calcium will activate this pathway. And can anyone tell me why insulin will activate this pathway? Like what's the
Activate or deactivate?
It will activate.
No, I'm asking. What did you say?
Insulin. Why would insulin activate this reaction? Okay, basically, do you have an answer?
Yes.
Yes.
Insulin promotes glycolysis. When you have glycolysis, you have pyruvate. When you have pyruvate, you do the electron transport chain. When you do the electron transport chain, you need this.
Yeah, basically. In other words, basically, what does insulin mean? Insulin means we have high glucose levels. High glucose levels means the body wants to use up this glucose to produce energy to decrease the amount of glucose. So high glucose means high energy, means energy production. Okay. Now we've reached the main point of the session, which is the citric acid cycle, which will also occur in the mitochondrial matrix. It's an anabolic pathway, which means it's both anabolic and catabolic. And anabolic means biosynthetic, which in the beginning we will take a 4-carbon compound, a 2-carbon compound, and produce a 6-carbon compound. So it's anabolic. Then, as we go on with the cycle, we will break down this 6-carbon compound into a 4-carbon compound, so it is catabolic. This cycle starts and ends with the same component, obviously, because it's a cycle, and it consists of 8 steps in total, with 3 regulatory / rate-limiting steps. The steps 1, 3, and 4, and we will explain why. Steps 1 to 4, the main aim of steps 1 to 4 is to acetyl groups of acetyl-CoA react with oxaloacetate, and 2 carbon dioxide molecules are liberated. Steps 5 to 8, oxaloacetate is regenerated. Okay. So this is the general reaction, or the general formula of the citric acid cycle. You will need to remember that 2 carbon dioxides are produced, 3 NADH, 1 FADH2, and we will go over each step so that you remember where it's produced, where it's used up. This cycle is crucial to energy production, of course, and this cycle intermediate, there's another function for the citric acid cycle, which is that the cycle intermediates are substrates in various biosynthetic reactions, which means that we will use them to produce other compounds, which will be explained later. Okay, first step of the citric acid cycle. So first of all, with any reaction, you need to remember 4 things. You need to remember the reactants, the products, the enzyme, and the type of reaction. So the first reaction is a condensation or addition of acetyl-CoA with oxaloacetate to produce citrate, a 6-carbon compound, by citrate synthase. So remember that this is a synthase, which is a lyase, and not synthetase, a ligase. What's the difference? Lyase will use water, and it utilizes this water to break bonds in the mechanism, and it will not require ATP, even though this is it's an anabolic reaction. It's biosynthetic, which usually requires energy, but since it's a lyase, it will not require energy. 6 carbons are produced. Both acetyl-CoA and oxaloacetate must be present to initiate the cycle, obviously because they are the substrates that are required. Acetyl-CoA plus oxaloacetate plus water produces citrate plus coenzyme A, which is released from the acetyl-CoA, and 1 hydrogen atom. Okay, so one issue with this chapter is that it's filled with mechanisms and detailed mechanisms, random amino acids, and we weren't sure whether we need to remember this or not. So my advice is to read it, and every time you revise, you go over it again, but don't memorize the actual mechanism. Just remember the amino acids involved, and remember whether it's an acid, a base, just generally. Citrate synthase is a homodimer. The active site is a cleft between 2 subunits. Oxaloacetate binds to the open form of the enzyme, and once it binds, it causes a conformational change, forming the closed form and forming the acetyl-CoA binding site. So after oxaloacetate binds, acetyl-CoA binds, and then the reaction can take place. The reaction, as I mentioned, this is a regulatory or rate limiting step. Why? Because it has negative delta G, or it produces energy. So why is energy released? Due to the hydrolysis of the high energy thioester bond of acetyl-CoA. There's a thioester bond, as I mentioned before. The thiol group carries an acetyl group, and once this bond is hydrolyzed, energy is released. And this is where we mentioned that it's a lyase and it will require water. Okay. Next, second step is isomerization of citrates to isocitrate by the enzyme aconitase, and it's called this name because there is a cis-aconitase intermediate. So why does this happen? Because isocitrate is a secondary alcohol which is more reactive and more easily oxidized in the next step than citrate, which is a tertiary alcohol. So we take the citrate that we produced and we isomerize it to another form of the same molecule, which is isocitrate, which is a secondary alcohol, and it's more easy to oxidize in the third step of the citric acid cycle. So this reaction has a positive delta G, which means that it requires energy, and usually the backwards reaction will be favored. But since here we continuously remove the product, which is isocitrate, the forward reaction is actually favored. And you can see that water is involved here, but since the water is released and then re-added, the net use of water is 0, therefore it's not counted as a reactant. And also, as you can see, I've highlighted the names of histidine and serine. You need to remember that these are the amino acids involved in this step. Okay, 2 questions and then Ahmed will continue with the citric acid cycle. What is the main driving force that makes the citrate synthase reaction highly favorable? Is it ATP hydrolysis, release of carbon dioxide, hydrolysis of the thioester bond in acetyl-CoA, or oxidation of NADH?
Is it C?
Yes, it's C, as I mentioned, because when we have hydrolysis, we will have energy release. When we have energy release, the forward reaction is favored. Okay. If oxaloacetate levels are very low, which step will be most directly effective? Yes.
Oh, me?
Yeah.
It's the first step, but it's not written here.
No.
It's not?
Okay. Oxaloacetate levels are low. What does this mean? Yes.
B.
Yes. Can you explain why?
First,
Yeah, this step.
Okay, sure. Give him one hand, it's fine. It's because oxaloacetate is a reactant in this reaction. So logically, when you don't have oxaloacetate, obviously the reaction cannot take place because you don't have the reactant. Yeah, so that's basically it for my part. I would also like to give you guys some mnemonics, but I'll do that after we're done with this session. Thank you.
So, good morning, guys. I'm going to continue after my colleague, but before I continue, I'm here to help you. So, if you don't understand anything or you have a question, please stop me. Okay? So, we're going to start with the third reaction, where isocitrate is converted into alpha-ketoglutarate. This reaction is an oxidative decarboxylation reaction. What do we mean with oxidative decarboxylation? It is two reactions in one, which is oxidation reaction and a decarboxylation, a removal of a carbon molecule. This reaction is catalyzed by isocitrate dehydrogenase, and this reaction is an important reaction. Why? Because this is the first link between the TCA, which is the citric acid cycle, and the ETC, which you are going to take later in the next chapter. So, the isocitrate dehydrogenase is the enzyme that catalyzes this reaction. We have three isoforms of this enzyme. First one, all of them are having the same name. Someone could ask, what is the difference between them? What does isoform mean? Isoform is the same enzyme, but in a different location, or uses a different coenzyme. So, we have three isoforms: isocitrate dehydrogenase 1, 2, and 3. 1 are found within the cytoplasm. 2 and 3 are found within the mitochondria. 1 and 2 uses NADP as a coenzyme, while 3 uses NAD and not NADP. This enzyme uses also manganese, the isocitrate dehydrogenase, uses manganese as a cofactor, and tyrosine is found in its active site. No need to know what is the function of the tyrosine or manganese, just know them, that they are there. Okay. The fourth reaction, we're going to convert, So in this reaction, we're going to convert the isocitrate into alpha-ketoglutarate dehydrogenase. We are going to remove a carbon in the form of CO2, as well as we're going to produce the first molecule of NADH. This molecule of NADH, this is what links the TCA to the ETC. Then the fourth reaction, we're going to convert the alpha-ketoglutarate into a 4-carbon molecule, which is the succinyl-CoA. The same reaction as the one before it. We have a 5-carbon molecule, we are going to convert it into a 4-carbon molecule, which is the removal of a carbon and the production of a second NADH. But here we have an exception. We are going to add a CoA instead of that carbon that we removed. So we're going to remove a carbon and add a CoA instead of it. As well as we're going to produce the second molecule of NADH. So till now we have 2 molecules of NADH removed, produced, and 2 CO2 molecules removed. Something you need to know, that the enzyme that catalyzes this reaction is called alpha-ketoglutarate dehydrogenase complex. And once we mention a complex, you know that this complex consists of many enzymes and many coenzymes. That is similar to the one my colleague mentioned, which is the pyruvate dehydrogenase complex. It has, of course, 3 enzymes and 5 coenzymes. The 3 enzymes are the alpha-ketoglutarate dehydrogenase, dihydrolipoyl transaminase, and lastly, the dihydrolipoyl dehydrogenase. Each enzyme does the same as my colleague mentioned. As well as I said that it has same cofactors or coenzymes, which is the TPP, CoA, lipoic acid, NAD, and FAD. Also, they do the same reactions as before. But there is a difference between this enzyme and the pyruvate dehydrogenase complex. This is the only difference, which is that this enzyme does not involve covalent modification. It only involves allosteric. It does not involve covalent modification. This is the only difference between both enzymes. Other thing you need to know that when we introduce the CoA, we bind the CoA to the succinate through a bond which is called the thioester bond. This thioester bond is a high energy bond. What do we mean with high energy bond? This means that upon breakage of this bond, the break of this bond are going to yield a high amount of energy. Of course, we don't need this energy to be lost, so we're going to conserve it. But how are we going to conserve it? We are going to know in the next reaction. So, which coenzyme in alpha ketoglutarate dehydrogenase decarboxylates alpha ketoglutarate?
C.
Is that D? TPP?
Okay.
Can you explain?
Okay. TPP is the coenzyme that my colleague mentioned that it decarboxylates and removes the carbon. It does the same action in the alpha ketoglutarate dehydrogenase. Is everything clear till now? Does anyone have any question? Okay. So in the fourth reaction, it was the end of phase 1, which was the oxidation of two molecules of CO2. Step number 5 is the beginning of phase 2, which is the regeneration of oxaloacetate. But during the regeneration of oxaloacetate, we can produce some energy molecules and reducing coenzymes. So in the fifth reaction, as I said, we are going to break the thioester bond. We're going to release a high amount of energy. This high amount of energy, we're going to conserve it. How? We're going to put it in an energy molecule, which is the GTP. We're going to break this bond. We're going to use the GTP or ADP, both are the same. Someone could ask what is the difference? Both are the same, but an enzyme may use ATP, other enzyme may use GTP. It depends on the enzyme specificity, but both are energy molecules. So we're going to break the bond, release the energy, add it to the GTP or ADP, and add the phosphate group. So after doing this, we will yield a succinate and ATP or GTP, as well as we are going to remove the CoA because we broke it. In this reaction, this enzyme, which is called succinyl CoA synthetase, this enzyme has a histidine in its active site, as well as we have two forms of the enzyme. Both does the same work, but the difference is what? Is that one is specific for ATP, while other is specific for GTP. But what if we produce GTP, but the cell needs ATP? That's okay. You're going to convert the GTP into ATP. How? We're going to remove a phosphate group, add it on the ADP to yield ATP and GTP. But what is the name of the enzyme that catalyzes this reaction? That is called nucleoside diphosphate kinase. This is the enzyme that catalyzes the transfer of phosphate from GTP to ADP. But what if the cell needs the GTP? We will produce the GTP. But what is the use of this GTP? The use of it, we use it for either DNA or RNA or protein synthesis. So in step number 6, we ended with succinate. Now we're going to convert the succinate into fumarate in an oxidation reaction. This reaction is unique. Why? Because the enzyme that catalyzes this reaction are not found within the mitochondrial matrix. It is found tightly bound to the inner mitochondrial membrane. Because as you know, the mitochondria consists of two membranes. All all of the citric acid cycle enzymes are found here, except the enzyme that catalyzes this reaction, which is the succinate dehydrogenase, are found here, tightly bound to the inner mitochondrial membrane. And also, this enzyme uses FAD instead of NAD. Why does it use FAD instead of NAD? Because FAD is a stronger oxidizing agent. What do we mean with a stronger oxidizing agent? That it has a higher reduction potential, which I'm going to explain now. This means that the FAD can take the hydrogen from the succinate faster than NAD. That's why we use FAD instead of NAD. So we are going to remove a hydrogen from here, remove a hydrogen from here. Now, I don't know if you know this, but the carbon can only do 4 covalent bonds. So here it has a bond with 2H and here with 2H, and one with this carbon and one with this carbon. So each carbon has its 4 bonds. So removing a hydrogen from here and removing a hydrogen from here, each one needs to have an extra bond in order to have its 4 bonds. So we are going to do a double bond here. So we remove the hydrogen, a hydrogen. We are going to replace these hydrogens with a bond, which is the double bond here. So now each carbon has 4 bonds. I think this is the only thing you need to know from here. So we're going to remove hydrogen, add them to the FAD, put a hydrogen bond, and produce a KDH2, and fumarate. And step number 7, it is an easy step. Fumarate is hydrated to form something called malate. So this is done by an enzyme called fumarase. Fumarase here, it is a lyase. What do we mean with a lyase? It adds a water molecule, but doesn't break the molecule into 2. It just breaks a double bond, converted into a single bond. So we're going to add a H2O to break this bond into a single bond, to produce malate. Now, for the last step is done to regenerate the oxaloacetate that we started with. So this is an oxidation reaction. Malate, we are going to convert the malate into oxaloacetate in an oxidation reaction. Here, a third molecule of NADH will be produced. So the enzyme that catalyzes this reaction is called malate dehydrogenase. We're going to take 2 hydrogen molecules from malate, add them to NAD to produce oxaloacetate and NADH. Easy. You need to know that histidine are found in the active site of this enzyme. And you need to know this enzyme because we have 2 isoforms also from it. One which is found in the mitochondrial matrix, while the other is found within the cytoplasm. So, which of the following enzymes of the citric acid cycle catalyzes the reduction of FAD to FADH2?
Succinate dehydrogenase.
Which of the following reaction involves a substrate level phosphorylation? لا خلاص حتى
B.
دي صح. Okay, so oxidation of which of the following substrates in the citric acid cycle is not coupled to the production of NADH or FADH2?
Malate.
لا. fumarate صح.
Can you go back?
Malate is converted into oxaloacetate in an oxidation reaction, so we produce NADH. Isoprate is converted into alpha ketoglutarate in an oxidative decarboxylation, so you produce a molecule of NADH. Alpha ketoglutarate is converted into succinyl-CoA in an oxidative decarboxylation, so you produce also NADH. Succinate is converted to fumarate in an oxidation reaction, but here you produce FADH2. Fumarate is the only one that is not, you don't produce or is not oxidized. Fumarate
Nothing
is
is produced, sir.
Huh?
Nothing is produced.
No reducing agents are produced in this reaction. So, in the citric acid cycle, when two acetyl-CoA are oxidized to CO2, the total yield of NADH is
D.
D. No.
C.
C. صح. So, what will be the answer if I said that two pyruvate molecules?
4. If what?
صح صح four, sorry. Okay. So, what are the keys of carbon atoms in the citric acid cycle? We condensed, we added the acetyl-CoA, which is the 2-carbon molecule, to the oxaloacetate, which is the 4-carbon molecule, to produce what? A 6-carbon molecule, which is the citrate. Now, the two molecules of CO2 that enter as acetyl-CoA are not the ones oxidized, because we oxidize 2 carbon molecules and we remove them. So the 2 carbon molecules that is removed are not of acetyl-CoA. They are of oxaloacetate. And this figure shows that. I'm not sure if you can see, but you can see here that the 2 carbon were the 2 of acetyl-CoA were the top 2. The top 2 remained untouched until the end of the reaction. So the 2 carbon molecules that oxidize during the reaction or during the citric acid cycle are the ones of oxaloacetate and not acetyl-CoA. Is it clear? Is it clear?
Yes.
So what is the net production of one molecule of pyruvate released from glycolysis and joining the citric acid cycle?
Me? D.
دي صح. سمعت خلاص منك. Okay. So how many carbon dioxide molecules are consumed in 2 terms of TCA cycle? لا من هو خلاص.
0.
صح 0. Why?
Because
I will not consume. We just consume.
Just leave it.
So, the citric acid cycle is an anabolic pathway. What do we mean with anabolic pathway? That is, it is associated with both catabolic and anabolic processes. Catabolic, that is obvious because we oxidize the 6 carbon molecule into 4 carbon molecules. So we remove 2 carbon molecules and we produce energy. This energy is conserved in the reducing agents, which is NADH and FADH2. Okay, this is the catabolic pathway. The anabolic pathway, that is important, and you need to know the examples, to memorize the examples, because you're going to get asked about it. So, anabolic pathway, we just use the citric acid cycle intermediates as precursors. Why? For the synthesis of other molecules. Is it understandable?
Yes.
Do you have any question?
No.
Okay. So, the first one and the most important one is the oxaloacetate. Why? Because oxaloacetate is used for gluconeogenesis. And you took that, by the way. Oxaloacetate, we can take the oxaloacetate from the citric acid cycle and convert it into pyruvate and continue gluconeogenesis by an enzyme, I think so, that is called, no, I forgot. Okay, other reaction for oxaloacetate is that we can use oxaloacetate for the synthesis of certain amino acids. Because the oxaloacetate is considered as an alpha keto acid. I think so. You took, you know what does alpha keto acid mean? What does it mean?
لا, I want to ask, I want to correct something.
Okay.
In gluconeogenesis, it's oxaloacetate to PEP.
But at the end you will end with pyruvate. But you're correct.
No gluconeogenesis, you make glucose.
Are you okay? You will convert oxaloacetate to phosphoryl pyruvate. Oh, okay.
No, you're right. Because you will produce glucose
glucose بعدين glucose goes through glycolysis to produce pyruvate.
But this is talking about
No,
gluconeogenesis.
no, he's correct, he's correct. That should be converted into phosphoenolpyruvate and then we should continue glycolysis.
But
Gluconeogenesis.
here it doesn't mean إنه oxalic acetate is directly converted to pyruvate. يعني it could have many steps. مثلا second one, oxalic acetate to certain amino acids through many steps. Yeah.
Okay.
That's how it's
You know what is an alpha keto acid? What is an alpha keto acid?
It's,
Hmm?
you take glutamine, I think, and then you progress to.
No.
No.
The structure of amino acid is that you have a carbon, a carboxyl group, a hydrogen, an amine group, and an R group. When you remove the NH3, when you remove the amino group, you still have this structure. This structure is referred to as the alpha keto acid. So, oxaloacetate and alpha ketoglutarate, both are considered as alpha keto acids. So, when we add an amine group to oxaloacetate or alpha ketoglutarate, we're going to convert them into amino acids. That's why they are used for the synthesis of certain amino acids, both oxaloacetate and alpha ketoglutarate. Oxaloacetate, what are the amino acids produced? Most prominent one is aspartate. Alpha ketoglutarate is converted into glutamate or glutamine, and there's more, but these are the most prominent examples. Then, the succinyl-CoA, isn't succinyl-CoA and citric acid cycle intermediate?
Yes.
So, we can use succinyl-CoA for the production of heme. What is heme? Heme is used for the synthesis of hemoglobin, which is a protein found in your blood. Lastly, if we have excess citrate, we can take this citrate outside from the mitochondria into the cytoplasm, break it down into oxaloacetate and acetyl-CoA, which are the precursors of citrate plasma. So, when we break it down into oxaloacetate and acetyl-CoA, we're going to take the acetyl-CoA for the synthesis of some fatty acids. Other two reactions, we can use oxaloacetate for the production of DNA or RNA bases. You know that RNA or DNA bases are either divided into pyrimidine or purine. Pyrimidine is the single ring, while purine is the double ring or two rings. Oxaloacetate is converted directly into pyrimidine bases. Okay, while alpha ketoglutarate is converted into purine but indirectly. How? First of all, as we said, alpha ketoglutarate is considered what? An alpha keto acid. So, when we add an amino group to alpha ketoglutarate, we're going to get glutamate. We will take this glutamate and convert it into purine bases, simply. Then we have some, before I explain what is anapyretic reactions, when we use all of the citric acid cycle intermediates to produce products such as heme, amino acids, or glucose, aren't the citric acid cycle intermediates going to finish? That is going to finish. So we need to replenish them. How do we replenish them? We replenish or regenerate the citric acid cycle intermediates through reactions called anapyretic reactions. Anapyretic reactions are reactions that regenerate the citric acid cycle intermediates. So, in this, what will happen if the oxaloacetate finished? We need to regenerate it. So, we can regenerate it from aspartate. Or, an important reaction, and this you need to memorize it. If acetyl-CoA is more than oxaloacetate, unequal ratio, acetyl-CoA is more than oxaloacetate, TCA will run normally until we don't have oxaloacetate. Why? Because we have acetyl-CoA more than oxaloacetate. So, we are going to stop the production of acetyl-CoA, inhibit the enzyme that produces acetyl-CoA, and activate the enzyme that produces oxaloacetate until we have equal amounts of both. So we can run the TCA normally. The enzyme that is responsible for the conversion of pyruvate to oxaloacetate is called, you took it.
What's the question?
what is the name of the enzyme? Okay, sorry, pyruvate carboxylase. So we're going to inhibit the pyruvate dehydrogenase complex, and we are going to activate the pyruvate carboxylase. Until we have equal amounts, we are going to stop this and activate the cellular. Here it is, pyruvate into acetyl-CoA. This is the normal reaction. When we have, when acetyl-CoA to oxaloacetate ratio is high, and it means that acetyl-CoA is more than oxaloacetate, we are going to convert the pyruvate into oxaloacetate. And we are going to inhibit this until we have equal amounts. Other reactions, like we have certain fatty acids, not all fatty acids. We have certain fatty acids that when we break them down, they are going to yield succinyl-CoA, which is an citric acid cycle intermediate. Last one is that we can reverse this reaction. Here, I said here aspartate and glutamate. We can reverse it. Convert the aspartate and glutamate into citric acid cycle intermediates, which is here. Aspartate, you can convert it into oxaloacetate. You don't need to know the name of the enzymes, they are not mentioned as well. And here, we can convert the glutamate into alpha-ketoglutarate to replenish or to regenerate the citric acid cycle intermediates. So, if there is pyruvate carboxylase deficiency or inhibition in liver cells, this will result in a decrease in the level of which of the following molecules? E, oxaloacetate. Yes, صح. Okay. Now, citrate metabolism. I said when we have excess citrate in the cell, we are going to take the citrate into the cytoplasm and break it down into its precursors. Okay. So we are going to continue with the citrate metabolism. I told you that when we have excess citrate in the cell, the mitochondria, we're going to take the citrate from the mitochondria into the cytoplasm. So imagine that this is the mitochondrial membrane. The membrane. Here is the cytoplasm. Here is the mitochondria. We are going to take the citrate from the mitochondria into the cytoplasm. I told you we're going to break it into two molecules. What are the two molecules?
Break what?
The citrate. Two molecules.
ADP and no,
oxaloacetate or acetyl-CoA.
We're going to break it into oxaloacetate and acetyl-CoA. Here we're going to use an enzyme called ATP citrate lyase. ATP citrate lyase. From its name, does it use ATP or no? Yes, it does. But here it uses the ATP as a source of energy and not as a source of phosphate. So we're going to convert ATP into ADP and then inorganic phosphate. The acetyl-CoA, we said that we are going to use it for the synthesis of what? Huh? Yeah. We're going to use the acetyl-CoA for the synthesis of fatty acids. Fatty acids synthesis. Oxaloacetate will be converted into malate. Remember in the citric acid cycle, the last step was malate into oxaloacetate. It was a reversible reaction. So we can convert the malate into oxaloacetate and the oxaloacetate into malate. So here we're going to convert the oxaloacetate into malate by the same enzyme, malate dehydrogenase. There in the citric acid cycle we produced an ADH, right? Here we're going to use an ADH. So we're going to produce an ADH and get an going to use NADH and get NAD. Now, the malate has two pathways. It will either enter to the mitochondria again, or it will be converted into pyruvate. But we're going to ask, which pathway should which one is going to be used? Which pathway? It depends on the cell need. Why? Because the malate, when we convert it into pyruvate, we're going to produce NADPH. We took NADPH, right? You know what is NADPH? It's a reducing agent. If the cell needs NADPH, the malate will be converted into pyruvate, and then the pyruvate will re-enter into the mitochondria. If the cell doesn't need NADPH, the malate will enter directly. Okay? Now, if the cell converted the malate into pyruvate, it means that it needs NADPH. Pyruvate, after that, will enter into the mitochondrial matrix. The pyruvate also has two pathways. It will either be converted into acetyl-CoA normally, or it will be converted into oxaloacetate. When will it be converted into oxaloacetate?
Maybe.
Huh? When the acetyl-CoA is more than oxaloacetate, we convert this pyruvate into oxaloacetate. Or into acetyl-CoA. Normally, if the pyruvate was converted into acetyl-CoA, we will continue the citric acid cycle normally. We will condense the oxaloacetate with acetyl-CoA to get citric.
Can you rotate it?
So this is the reaction. This is the citrate metabolism. So as you took a brief knowledge about the citrate metabolism, I'm going to repeat it fast. If we have excess citrate, the citrate will be transferred into the cytoplasm, will be hydrolyzed into its precursors, which is the oxaloacetate and acetyl-CoA, by ATP citrate ligase. This enzyme uses ATP as a source of energy. That's why you have ADP and inorganic phosphate. The acetyl-CoA, which is released, which will be used for fatty acid synthesis, while oxaloacetate will be converted into malate by malate dehydrogenase. We will use NADH. We will not produce one, we will use one. The malate will either enter to the mitochondria or will be converted into pyruvate. Which pathway will be used depends on the cell need. If the cell needs NADPH, it will convert the malate into pyruvate and produce NADPH. The pyruvate will enter into the mitochondria. Pyruvate has two pathways also. Will either be converted to oxaloacetate or into acetyl-CoA. It depends on which one the cell needs more. So here is the most important question, and we had a question on this in the summer. Acetyl-CoA molecules can be used in biosynthetic pathways such as fatty acid synthesis. Okay. Which of the following enzymes are found within the cytoplasm and not in the mitochondria?
Malate dehydrogenase Malak. Two.
No, that's fundamental.
عملية الهيدروجينيز هنا لسه بعرف يستخدم الأوكسالوأسيتيت.
دي answer.
ATP citric ligase. صح.
And
أنت قلت دي؟ Okay, so صح. A and C. Malate enzyme and ATP citric ligase. Both are found here in the cytoplasm, but are not found within the mitochondria. What are the products formed from the action of ATP citric ligase? تجاوبت كثير خلاص. بعد
Is it A?
لا
ده كده.
صح.
C.
سي acetyl-CoA and oxaloacetate صح. We came to the regulation of the citric acid cycle.
Who wants to answer?
What's else?
Okay, so the regulation of the citric acid cycle. As my colleague Linah mentioned, the regulatory steps are the reaction number 1, 3, and 4. Okay, so just before we start the regulation, this is a point you need to know. That any enzyme, or most enzymes, are regulated either by their product concentration or by their substrate concentration. If we have a high concentration of the substrate, will the enzyme be inhibited or activated? Why?
Because it wants the product.
Yes, we need to get rid of the substrate. But what if we have a high concentration of the product?
Inhibit.
Inhibited. Because we have enough amount, we don't need more. So we stop the enzyme. So for citrate synthase and isocitrate dehydrogenase, both are regulated by ATP to ADP ratio as well as NADH to NAD ratio. What do you mean with this? That if the ATP is more than ADP, does this mean that the cell has high amount of energy or low?
High.
High. So do we need to activate these enzymes to produce energy?
No.
No. So we inhibit the enzyme. Vice versa for ADP. If ADP is more than ATP, does this mean that the cell has high amount of energy or low?
Low.
Low. So are we going to activate or inhibit the enzyme?
Activate.
Activate. Why?
We need energy.
To produce energy. You have to add some points that citrate also inhibits citrate synthase, as I said, because it is a product of this enzyme. So when we have a high amount of citrate, we are going to inhibit this enzyme. Another point that succinyl-CoA, if succinyl-CoA is high in the cell, it is going to inhibit this enzyme. Why? I think you took it, which is the feedback inhibition. Feedback inhibition, that a molecule later in the cycle or the pathway inhibits an enzyme in the beginning of the cycle or the pathway. The same thing for isocitrate dehydrogenase. Lastly, alpha ketoglutarate dehydrogenase are also activated and inhibited by the same manner, but here it is ATP to ADP, ATP to AMP. Here it was ATP to ADP, here it is ATP to AMP. The same manner, the same concept, but ATP to AMP, this is the only difference. And lastly, it is also inhibited by a high concentration of its product, which is the succinyl-CoA. Because alpha ketoglutarate dehydrogenase converts the alpha ketoglutarate into succinyl-CoA. So when succinyl-CoA, which is the product, increase, it is going to inhibit the enzyme. Lastly, the calcium regulation. You don't need to know all of this, but what you need to know that when the calcium level in the matrix increase, it is going to activate all the citric acid cycle enzymes. So when calcium increases in the mitochondrial matrix, it is going to activate citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase. Citrate synthase is the enzyme that catalyzes the condensation of acetyl-CoA and oxaloacetate to citrate. Which of the following is an activator of this enzyme? Again?
A.
A, صح, ADP. Why?
Because then you have low energy.
Sign of low energy.
صح. Is everything clear?
Yes.
Do you have any questions? No? Okay. Which statement concerning regulation of citric acid cycle is true? No. صح. Because high levels of ATP indicates that we have high amount of energy. So it is going to inhibit the reactions of the cycle. The same for NADH. High levels means that we have high amount of energy. So we're going to inhibit the reactions of the cycle. Low levels of ATP indicates that we have low energy. So we're going to activate the reactions of the cycle. The same for high levels of ADP. Means we have low energy, so we activate the cycle. Which of the following substances, when present in high concentration, inhibits the alpha ketoglutarate dehydrogenase complex?
C.
C, صح. Why?
Because it's the product. If you have too much, you don't need to make more.
Okay, lastly, the citric acid cycle and the human disease. if we have mutation in some of the citric acid cycle enzymes, or if we have mutation in the genes that produce some of the citric acid cycle enzymes, we're going to have diseases. One of these diseases is the encephalopathies, which are diseases related to the brain. So if we have a mutation in the genes that produces alpha-ketoglutarate dehydrogenase, succinate dehydrogenase, fumarase, and succinyl-CoA synthetase, we will have encephalopathy. There are more diseases, but the one mentioned in your PowerPoint was only this.